This is going to be your video recap of the light-independent reactions, also known as the Calvin cycle. So first of all, let's notice and remind ourselves that we are still within the chloroplast of the cell. So our chloroplast structure is given over here on the left, and this is where all of photosynthesis is taking place. Now, the light-dependent reactions that we learned previously, remember that was in the membrane of the thylakoid, but now we are focusing on this cycle right here, and that is just happening in the liquid, and that liquid, remember, is called the stroma. So we're in the liquid of the chloroplast. We are no longer relying on that thylakoid membrane. You'll notice that this is a little bit different from the light-dependent reactions in that this is really a biochemical cycle. This is not the movement of things through an electron transport chain. We're not talking about membranes or concentration gradients or any of those sorts of things. We are really focusing on the molecules and how they are being rearranged in different parts of the cycle. So speaking of the molecules, I am going to start out by just sketching a general depiction of each molecule in here. You don't need to know all of the details of these molecules, but sometimes I think it's helpful to have a representation of them here. So we're going to start out with a 5-carbon molecule, and there's actually 3 of those. It's going to be built into a 6-carbon molecule. Still 3 of them. That 6-carbon molecule is going to be split in half. Okay, so 2 separate 3-carbon molecules. So 3 of each. And then 1 of those molecules will leave the cycle altogether, leaving the remaining 5 molecules to stay in the cycle and be regenerated. So I wanted to go ahead and make sure those molecules were all represented in this diagram before we got started. Now, our first step here is we have to have the input of carbon because the reactants of photosynthesis, remember, include carbon dioxide. Carbon dioxide was not used in the light-dependent reactions. So here we have the input of CO2, and that is coming from the atmosphere. Plants have openings in their leaves that allow CO2 to come into the leaf and into the chloroplast. So this CO2 has a carbon atom in it. Remember, we started with a 5-carbon molecule, 3 of them, and now we have 6 carbon molecules. Well, that carbon from the CO2 is getting added. So 3 molecules of CO2 are combining with these 3 molecules of our starting material to make 3 molecules of this 6-carbon compound here. So that 6-carbon compound isn't glucose quite yet. It's a different compound. So we still need to go through a few more steps in order to generate glucose. Those steps are going to involve rearranging this molecule and splitting it apart. And that is going to require us to put in some ATP. So it requires the input of energy. And you can see that here with our ATP. When we input ATP, that is going to give us an output of ADP and the inorganic phosphate. Now notice what we're doing to this molecule. We're splitting it in half. So that means we also need some electrons because we're going to need to make sure that all of the valence shells of the new molecules are filled with electrons. So if you remember from the light-dependent reactions, that produced NADPH, which is carrying electrons. Those electrons get dropped off in the Calvin cycle, and then the electron carrier leaves minus its electrons, So now it is in the form of NADP+. Now that is reusable, so it can go back to the light-dependent reactions, and it can pick up some more electrons, and it can keep going back and forth, shuttling those electrons with it. So what we have now brings us to step 2. So step 2 is really describing everything that happened between step 1 and this point. So if we look at what we did, all that happened was we input energy and electrons to rearrange organic molecules, right? We have the input of ATP, we have the input of electrons, and we have rearranged molecules. So I'm going to summarize this as input of energy and electrons to rearrange molecules. So again, you are never going to have to say on a test how these molecules are rearranged. I just really want to make sure that you understand that that's the basis of what's happening, but not get bogged down in the detail. So now that brings us to step 3. I think step 3 is pretty obvious from the diagram what's happening, because we have this arrow leaving the cycle and it's showing a 3-carbon compound leaving. And that's exactly what it is for step 3. 1 3-carbon molecule leaves the cycle. Now notice that is going to leave us with 5 more 3-carbon molecules. We had 6 total. We lost 1. The other 5 stay in the cycle, and they are going to get rearranged back into the starting material. That's our step 4. Now that rearrangement of molecules, just like we had at the top part of our cycle, that is going to require the input of ATP. So I will note that here. We're going to have the input of ATP to make that happen, and then once it's used, it's going to release the ADP plus inorganic phosphate. So now what we are left with is a description of what has happened with one cycle. This cycle has happened once. We've had the production of 1 3-carbon molecule. So if you remember the formula for glucose, it has 6 carbons. So we need this cycle to happen a second time. That way we will get a second 3-carbon molecule, and those 2 3-carbon molecules will combine to make glucose. So I'm going to make a note of that over here. So this cycle is going to happen 2 times, and then 2 3-carbon molecules equals glucose. So 2 of those 3-carbon molecules get joined together. That forms our molecule of glucose, and that is our final product of photosynthesis.